Okay, so welcome to this next video on antibiotics which target bacterial cell wall biosynthesis. Okay, so far in this biosynthesis pathway, we've seen how we assemble these N-acetylmuramyl pentapeptide with N-acetylglucosamine bound to it. These complexes, we assemble them bound to bactoprenol here by two phosphate groups, and currently it's on the inner leaflet of the phospholipid bilayer. So let's turn over the page and see what happens next. So what's going to happen next is that bactoprenol is basically going to move from being on the inner leaflet of the phospholipid bilayer to being on the outer leaflet of the phospholipid bilayer. So this is bactoprenol here, okay? And what's going to happen apart from that, it's still going to remain bound to the N-acetylmuramyl pentapeptide with the N-acetylglucosamine. But that's going to be short-lived, basically. Very soon, the N-acetylmuramyl pentapeptide with the N-acetylglucosamine is going to move. So let's draw this out. So here is the N-acetylmuramyl pentapeptide here. So here's the six-membered ring. Down here is the pentapeptide bit with the pyruval, which now I'll just draw as a box, basically. Okay, and here's the N-acetyl portion, which again I'll just draw as a box. And now it's linked to N-acetylglucosamine, which is this other six-membered ring here, with this fifth carbon going up here. And again, N-acetylglucosamine has an acetyl group on its amino group down here, but again I'll just draw that as a box. So here what we have now done is we have transported our monomer of the peptidoglycan cell wall onto the outer leaflet of the cell membrane, basically. So now what I need to discuss with you is the structure of peptidoglycan. Well, peptidoglycan is just basically a polymer of these structures here. So basically it's a polymer of N-acetylglucosamine with N-acetylmuramyl pentapeptide. So what you have is these N-acetylglucosamine molecules, so like this. So this six-membered carbon ring with an acetyl group on the amino group down here. And then it's bonded to this N-acetylmuramyl pentapeptide here, which again is this six-membered carbon ring, now with this pyruval and then the pentapeptide structure here, and also an acetyl group of the amino group down there, and the sixth carbon here. And then you just continue this pattern. You have another N-acetylglucosamine here, like so. So here's the six-membered carbon ring with that oxygen, and then the acetyl group of the amino group. So you alternate between having N-acetylglucosamine and N-acetylmuramyl pentapeptide, basically. So this is the N-acetylmuramyl and then pentapeptide. Okay, so you alternate between these two, and that basically is the structure of peptidoglycan. So what's going to happen to this monomer of this peptidoglycan that we have transported onto the outer leaflet of the cell membrane? Well, basically what you're going to do is you're going to cleave this bond here, and you are going to bind, what you're going to do is cleave that bond, and you're going to bind this monomer to the carbon of this next, of the free end of this polymer of peptidoglycan, basically. So you're going to extend the polymer. So if this is the free end of your polymer, so the rest of the peptidoglycan continues on this way. This was the free end. What you're now going to do is transfer this monomer onto that free end. And the enzyme which does that transfer of this monomer onto the free end of this peptidoglycan is called peptidoglycan transglycosylase. So this is peptidoglycan transglycosylase. Transglycosylase. Okay, so there is this enzyme which transfers this N-acetylglucosamine N-acetylmuramyl pentapeptide unit onto the growing peptidoglycan polymer. So this is the peptidoglycan polymer here. Okay, and of course this will be on the surface of the cell membrane. So if this is the phospholipid bilayer of the cell membrane here, then that picture looks about right. Okay, so you are taking these N-acetylglucosamine, N-acetylmuramyl pentapeptide units onto the outer leaflet of the phospholipid bilayer through this bactoprenol molecule, and then you are using the enzyme peptidoglycan transglycosylase to add that onto the growing peptidoglycan polymer, which is this polymer which is an alternation between N-acetylglucosamine and then N-acetylmuramyl pentapeptide, basically. Okay, right, so let's talk about our next antibiotic which can affect this. Basically, what happens to the bactoprenol afterwards? So once you've moved this unit, once you've moved this N-acetylglucosamine, N-acetylmuramyl pentapeptide off of the bactoprenol, what is left over is you have bactoprenol now bonded to two phosphate groups. So this is called bactoprenol pyrophosphate. Now, you have only a certain amount of bactoprenol in your cell membrane. So, if you want to continue transporting N-acetylglucosamine, N-acetylmuramyl pentapeptide units from the inner leaflet to the outer leaflet, you need to regenerate this bactoprenol. It needs to move back into the inner leaflet, and also it needs to do away with one of these phosphate groups. Remember, the original molecule that our UDP N-acetylmuramyl pentapeptide bound to was just bactoprenol bound to a single phosphate called bactoprenol phosphate, basically. So this was bactoprenol phosphate. So basically, you need to regenerate the bactoprenol phosphate from the bactoprenol pyrophosphate. And there is an antibiotic which prevents this movement of the bactoprenol pyrophosphate back onto the inner leaflet of the phospholipid bilayer, because basically what it does is it binds to the bactoprenol here, and that's the best I can draw of it binding. So it basically binds to it and keeps it on the outer leaflet. So once the bactoprenol pyrophosphate has been made, this antibiotic will bind to it and it will stop it from going back into the inner leaflet of the phospholipid bilayer, and it will stop it from cleaving off that second phosphate group. This antibiotic which does this is Bacitracin. And by stopping the regeneration of the bactoprenol phosphate, Bacitracin stops peptidoglycan synthesis, basically, because if you can't move the N-acetylglucosamine, N-acetylmuramyl pentapeptide units from the inner leaflet to the outer leaflet, then you cannot move the monomers which you're going to make the peptidoglycan out of, the units which you're going to use to make the peptidoglycan. You cannot move them from the inner leaflet to the outer leaflet, and therefore you cannot continue to synthesize peptidoglycan. So this antibiotic, Bacitracin, actually will completely halt peptidoglycan synthesis. Right, okay. So let's discuss the next portion of this pathway. So we're now going to turn our attention basically to transpeptidases. So let's say we have synthesized some peptidoglycans. So we've synthesized these polymers of N-acetylglucosamine, N-acetylmuramyl pentapeptide units, basically. Okay, now let's say we have multiple of these polymers stacked on top of one another. So let me show this. So you're not just going to make one polymer of peptidoglycan. That would be a bit useless, wouldn't it? There's no point just making one of these. What you have to do is basically make multiple of them and sort of thread them all together, basically, to make a strong structure. Okay, and I can't draw too much of this because I haven't got enough time. But what you need to do is you need to make multiple peptidoglycan strands, and you need to join them together in order to make a meshwork, basically, which is going to be the peptidoglycan cell wall. So here's our N-acetylglucosamine. This is an N-acetylmuramyl pentapeptide here. This is also, no, that's an N-acetylglucosamine next. Okay, so now let's draw the other peptidoglycan strand underneath, basically. Or, how else should we draw it? Well, it's difficult to show, actually. Let's draw another one, but it's easiest to show this, basically, when the two are sort of oppositely oriented. So I'm going to orientate them oppositely, basically. So let's have this one with its groups sticking out like so. Okay, so here is our N-acetylglucosamine, and I've drawn that on the... Have I drawn that on the wrong one? Yes, I think I have. So let's put that one on there. Okay, and we will... So this is the third carbon here, fourth, fifth. So then that will be linked up here, like so, and then you'll have the sixth one coming off up here, and then this one will have the pentapeptide, the pyruval and the pentapeptide group here, and then you'll continue on basically like that. What you can do is you can link up the pentapeptide group of this N-acetylmuramic acid with the pentapeptide group of this N-acetylmuramic acid. So you can link up the pentapeptide groups between the peptidoglycan strands. So these are two different peptidoglycan strands. Okay, and basically if we look back at our original structure for these pentapeptides, this structure which we drew out long ago, what you do is you take this amino group of this third amino acid of the N-acetylmuramyl pentapeptide. So this third amino acid here, which is either diaminopimedic acid in the case of Escherichia coli or L-lysine in the case of Staphylococcus aureus. Regardless, it has this amino group, basically. Now, what you do is you combine the amino group of one pentapeptide to this terminal carboxyl group of the entire pentapeptide, basically, and you can form a peptide or an amide link between the two. So, basically, if we just want to draw that simply, we can draw one of these pentapeptides having its carboxyl terminal here. So here's the carboxyl terminal, and another one here having its amino group coming off here. Okay, and this is getting a little crowded. I'll draw it here, even though that's incorrect. So here's the amino group, and basically what you're going to do is you're going to form an amide link between them. So the important thing is that you get links between these pentapeptide groups of the N-acetylmuramyl pentapeptide. Right, okay, and the enzyme which goes about forging these links between the pentapeptides is something called peptidoglycan transpeptidase. Peptidoglycan transpeptidase. It's also sometimes called penicillin binding protein, transpeptidase. The name of peptidoglycan transpeptidase is more truthful to what this enzyme actually does, but you will see it occasionally referred to as penicillin binding protein. Okay, and basically there are a number of antibiotics which work by inhibiting this peptidoglycan transpeptidase, because if you inhibit this enzyme, then you don't get the formation of these amide links between the pentapeptide groups of the N-acetylmuramyl pentapeptide component of the peptidoglycan strands. And basically what that means is that all the peptidoglycan strands are just uncross-linked, basically. So you just make loads and loads of strands, like so, where they're not cross-linked together, and that's useless. That's not going to create you a rigid structure. You've just made loads of strands pointlessly. So in order to create a rigid structure, what you need is loads of cross-links between these strands, basically, to hold it all together into one macroscopic structure. Okay, and that makes you a hard and strong and rigid cell wall. So unless you have the functional peptidoglycan transpeptidase, the final portion of this synthesis of this peptidoglycan cell wall doesn't work. So there are a number of antibiotics which inhibit this peptidoglycan transpeptidase, and they are by the name of the beta-lactam antibiotics. So beta-lactam antibiotics. Okay, and I will talk about the 4 different classes of beta-lactam antibiotics in just a moment. Okay, so there are absolutely loads of beta-lactam antibiotics in the world. And shall we move on to another piece of paper to discuss this? Okay, so there are absolutely loads of beta-lactam antibiotics in the world. In fact, I don't need a new piece of paper to do this. Okay, so for the 3rd time, there are absolutely loads of beta-lactam antibiotics in the world, and they can be categorized into 4 main families. The first and the most famous family of beta-lactam antibiotics is a family known as the penicillins. So penicillin is not just one drug. There are absolutely loads of drugs which are all considered to be penicillins. Regardless, people will say, I'm on penicillin. What they really will be on is a penicillin. So they might be on something like flucloxacillin, but that is a penicillin. So there are many, many different drugs which are all put under this heading of penicillin. Okay, and we'll have a whole video on the different types of penicillin. Right, so the second major family is something known as cephalosporins, or cephalosporins. I don't know how you're meant to pronounce that. Cephalosporins, I think, is better. So cephalosporins, again, it's not just one drug. It's loads of different drugs which are all put into this family of cephalosporins. Okay, next family, the monobactams. So monobactams, there are again many different drugs which are all categorized into this family of monobactams. And finally, there is the family carbapenems. So carbapenems is the 4th family of beta-lactam antibiotics. So all 4 of these are beta-lactam antibiotics. And again, they are not just one antibiotic, they are an entire family of antibiotics which are all classified as either being penicillins, cephalosporins, monobactams, or carbapenems. Okay, right. So the final antibiotic then that I want to talk about is, and I just want to say I've spelt carbapenems wrong. It should be carbapenems. Right. Okay, the final antibiotic I want to talk about is an antibiotic known as vancomycin, and I don't think any talk of peptidoglycan synthesis antibiotics would be complete without mentioning peptidoglycan. Vancomycin. Vancomycin basically binds to these pentapeptide groups of the N-acetyl-muramyl pentapeptide unit of the peptidoglycan, and it basically does this. It binds to the 2 dealanine groups, like so. So it binds over these 2 dealanine groups. And if you remember what I told you about the peptidoglycan transpeptidase reaction, what you do is you use the carboxyl group of that terminal dealanine here. You use this carboxyl group of this final dealanine that you have in the pentapeptide in order to link it to this amino group of this 3rd amino acid in the pentapeptide. So if you've got vancomycin bound over this dealanine group of the pentapeptide, like so, then you are not going to be able to link that carboxyl group of this dealanine group to the amino group of the 3rd amino acid of your pentapeptide, basically. And that is what this drug vancomycin does. It binds to the dealanine group and prevents the transpeptidase reaction, therefore preventing the formation of crosslinks between the peptidoglycan strands and therefore preventing the formation of a rigid peptidoglycan cell wall. So, if you don't have a rigid peptidoglycan cell wall, what's going to happen? Well, you're going to get osmotic lysis, because remember, in the first video in this playlist on antibiotics and antibiotic resistance, we discussed how the need for the cell wall is to prevent osmotic lysis, because the cytoplasm of the cell is hypertonic. It's got a much higher solute concentration than the extracellular fluid, so you will tend to get movement of water into the cytoplasm via osmosis, and this will raise the volume of that cytoplasm until the cell membrane bursts, unless you have a rigid cell wall structure holding everything together and preventing the rise in volume of the cytoplasm.